Hello everyone and welcome back to microbiology. In this chapter we're going to talk about microbial growth. You'll notice that we will do chapter 6 first and then we'll go to chapter 5. The reason for that is because we're actually working with this media actively in lab class right now, so I wanted to talk about this media first and then we'll talk about the metabolism in chapter 5. Your next exam will cover chapters 5, 6, and 7, and that's going to happen right before we go on spring break. So without further ado, let's go ahead and jump into this. So microbial growth, when we talk about microbial growth taking place, we're actually referring to increase into the number of cells, not the actual cell size. So when we talk about you growing, we're talking about you getting bigger, you getting larger. Where for bacteria, we're really just talking about the populations or the number of cells increasing and the number of colonies increasing. And you all have been able to witness microbial growth that happens on your plates when we did our kind of toe fungus, and when we did our very first labs, our environmental testing or swabbing of different places, we were noticing that the colonies increased on the bacteria, and the longer you let it sit, to a point in the incubator, we saw that more colonies were present. Now, in order for growth to take place, there are a couple of different requirements that have to be adhered to. We have physical and chemical requirements. Physical requirements are things like temperature, pH, and osmotic pressure, how salty something is, how much water is present, or I shouldn't say salty, but how many of the solute particles are present or absent. Chemical requirements are things that the bacteria will require to undergo metabolism. So carbon, nitrogen, sulfur, phosphorus, oxygen for energy, and even organic growth factors. So the first class of requirements that we're going to talk about are physical requirements. As you're probably already very intimately aware of and understand that there is a minimum temperature at which different species of bacteria, because not all bacteria require the same temperature, that has to be adhered to for the bacteria to grow. Optimally, the bacteria will have the greatest amount of growth and they'll flourish the most if we have the best temperature. And as I said before, these ranges are going to change depending on the species of bacteria that we're talking about. And then your maximum growth temperature is the highest temperature at which that bacteria can stand and it can continue to grow. If the temperature is too high, then it will cause the proteins of the bacteria to denature or unfold. And if the proteins denature or unfold and they lose their form, they're going to also lose their function and the bacteria will not be able to undergo its normal metabolism. So here is a handy-dandy slide that would be great discussion for a fill-in-the-blank question on your upcoming test here, is that, as I said before, not all bacteria require the same temperature as a growth requirement. We have what we consider to be here hyperthermophiles. Hyperthermophiles, they like a very high temperature. The optimum growth temperature for hyperthermophiles is about 90, and we're talking about Celsius here, so it's about 93, 94 degrees Celsius is the optimal growth temperature for these guys here. When you think about extremophiles, think about hyperthermophiles. They like really, really hot temperatures, almost boiling water hot temperature. Thermophiles will like temperatures that are about 59 to 60 degrees, or 60 to 61 degrees Celsius. So they like warmer temperatures than what we would normally see. Keeping in mind this is in Celsius, so if your body temperature is in Celsius, 37 degrees Celsius is about normal body temperature, and in Fahrenheit it's like 98, 99 degrees. That's something that's 60 degrees Celsius is pretty darn hot. Mesophiles, which are most of the bacteria that we work with are in this category of mesophiles. They like things that are right at about, boom, look at that, body temperature. So most organisms that we work with are mesophiles in our lab class, and then also most organisms that are pathogenic that cause disease in your body are mesophiles. Psychrotrophs are going to like a little lower temperature. It's kind of refrigerator temperature at about 24 degrees Celsius, and psychrophiles kind of fit in that same category as the hyperthermophiles or the extremophiles. They like really lower, much lower temperatures. So psychrotrophs grow at refrigerator temperatures between 0 degrees Celsius at the minimum growth range and about 30 degrees Celsius at the maximum growth range. We say that these guys can cause food spoilage because they like refrigerator temperatures. So if you have your peas in the back of the refrigerator that you forgot about, refrigeration doesn't kill microbes, it just retards or slows their growth. You got some peas that have a little fur growing on them back there. They're kind of spoiling, they kind of smell weird. They're probably psychrotropic bacteria that are causing that to happen. For food preservation temperatures here, notice that temperatures that are in this range above 60 degrees Celsius or above 140 degrees Fahrenheit, so just as a comparison, we have the Fahrenheit and the Celsius, you can kind of have an idea of what temperatures we're talking about here. Most of the time, most bacteria don't grow here. So unless it's a hyperthermophile, it's not going to grow at this area. Anything lower than 0 degrees Celsius or lower than the 35 degrees Fahrenheit. Also, you have very slow bacterial growth. It kind of slows it down or retards it, and anything lower than that, freezing, you really don't have a whole lot of growth that's happening there. Now, however, in this area here between 60 degrees Fahrenheit and about 15 degrees Celsius and about 52 degrees Fahrenheit, or about 130 degrees Fahrenheit, so we consider our dangerous zone. So this is when many bacteria can have rapid growth and can produce a lot of toxins and pathogens, and this is where we can get our food poisoning. So it's best, from looking at that graph and knowing what you know about food poisoning, if you've ever had it, it is not a delightful experience. That's one of the reasons why I have a great uncle, a grunkle, if you will, who refuses to eat leftovers. And I think he's had a couple of really bad bouts of food poisoning that he's just like, I'm not eating leftovers. So when you are trying to preserve your food or preserve your leftovers, you want to make sure that they get down to refrigerator temperature as quickly as possible. And the larger the surface area of your dish or your pan where the refrigerator air can get to every part of that dish, the better it is. So it's much more, you're less likely to have food poisoning if you take your leftovers and you put them in a series of smaller containers that have a larger surface area that will allow for the temperature of the food to get down to refrigerator temperature faster. We don't want to spend a whole lot of time in this danger zone, if you will. So notice that for this blue dish that's wider, greater surface area, more of it can get to refrigerator temperature faster. Notice how we're only spending, what, about an hour, hour and a half in this danger zone. Whereas with this larger dish here, that when we get it down, to refrigerator temperature is taking a little bit longer time. And in fact, we're spending, let's see, in the danger zone, we're 1, 2, 3, 4, 5 hours we're spending in the danger zone, and we're still not quite out yet. So at this point, this is the optimal temperature range for bacteria to replicate and to make more of themselves, so we can increase the number of bacteria that are in this dish because it's larger, it's deeper, and the refrigerator air can't get to all of the parts of it as quickly, so we end up having more a greater proliferation of bacteria in this dish. I've explained this to my husband, and he's finally, you know, agreed with me on this. If you would make spaghetti for dinner, he's like, oh, what's the point of putting it in a Tupperware dish? Let's just throw the whole thing in the refrigerator, because I can come back in a couple of hours, and if I dig in the middle of it, it's still warm. That's not a good thing. We don't want it to still be warm after you come back after a couple of hours and it's been in the refrigerator. Unfortunately, he hasn't had any bouts of food poisoning that I know of, and none of us have died or had horrible bouts of food poisoning. But you want to make sure that your food gets to refrigerator temperature, all of it, as quickly as possible. pH is another of our physical growth factors. Remember, just like with temperature, not all bacteria like the same pH, not all bacteria like the same temperature. Most bacteria like a pH of between 6.5 and 7.5, and remember, your pH scale goes from 1 to 14, where 7 is considered a neutral, and anything less than 7 is considered an acid. and anything above 7 is considered a base. So being that bacteria like pHs of about 6.5 to 7.5, they kind of hang around this neutral area here. So they kind of hang around here in most bacteria, especially those that are pathogenic. Molds and yeast like a slightly acidic pH of about 5 or a 6. So molds and yeast kind of hang out here. And then anything that likes pHs that are really, really acidic, those will also be extremophiles. We call those acidophiles. However, in your stomach, you have what's called Helicobacter pylori, and your stomach pH is about a 1, and it really likes that. So it's considered an acidophile, even though it's not an extremophile. So it just likes that really acidic pH. Osmotic pressure, or the concentration of solutes, salt or sugar solutes there. Different bacteria have different tolerances or different things that they like for osmotic pressure. A hypertonic environment or an increase in salt or sugar causes plasmolysis, which means that the cell wall will shrivel up and pull away and shrink. And cause the bacteria to die. Some bacteria like things that are really salty. We call them extreme or obligate halophiles, which means they need to have a high osmotic pressure, need to have a very high salt or sugar content, some sort of high salt content on there, very little water there. And then we have those that are called facultative halophiles, that even though they don't necessarily like a high osmotic pressure, they can tolerate or deal with it. So here we have a cell that's in an isotonic solution. So, and this is an idea that we've talked about before in Chapter 4, referred to the cell wall and the plasma membrane. That isotonic solution means that the concentration of water and solutes, or water and salt, are equal outside the cell and inside the cell. If a cell is in a hypertonic solution and it can't tolerate that high salt concentration, then water is going to move from an area of low concentration, where it's about 99% water inside the cell and only 90% water outside of it. So water is going to move out and causes the cell to plasmolyze, or to pull away from the cell wall and shrivel up and die. So now on to our chemical requirements. So the first chemical that we're going to talk about is carbon. Carbon is a very versatile structural organic compound. In fact, anything that contains carbon is considered to be organic. So if you're into sci-fi pics like I am, you've probably heard aliens refer to humans or other Terran or Earth species as being, carbon-based life forms. You are, in fact, a carbon-based life form. Carbon is an organic molecule that many living things can use as an energy source. All of the things that are considered to be organic molecules, proteins, lipids, carbohydrates, nucleic acids, they all have carbon as their backbone. Chemoheterotrophs, and this is an excellent fill-in-the-blank question. Chemoheterotrophs means that they use this chemical of carbon, in this particular case, not always carbon, it can be something different, but it uses this chemical and it eats other stuff. That's what hetero means, is other, feeds. So chemical feeds, or other feeding. So a chemical heterotroph means that it can use a source of chemicals that it has to eat from the outside. Auto means self. So if you drive an automatic car, it automatically changes from one gear to the next. If you drive a manual car, you have to shift gears when you go from one speed to the next speed. Autotrophs can feed themselves. They'll also use carbon, but they'll use the carbon in the form of carbon dioxide. So there are some bacteria, as we talked about in Chapter 4, that can undergo photosynthesis like plants, and they would be considered an autotroph. If a bacteria cannot undergo photosynthesis and it has to use something like, say, carbon in the form of glucose to get energy, it's considered a chemoheterotroph. It had to eat something else in order to meet its chemical requirements. Nitrogen is another chemical requirement. We find it in amino acids, and I'm going to start starring some of these things because I think they're very important. We find it in amino acids and proteins. Most bacteria can decompose or break down different types of proteins. One test that we have for that we are going to do is called a SIM test for sulfur, indole, and motility. With the indole test, we're looking to see if bacteria can use that very specific protein of indole. Some bacteria can decompose that protein, some cannot, and that's kind of the basis of a lot of our biochemical tests, whether or not this bacteria can use this chemical that's there, because they all can't, and that's how we're able to differentiate one species from another, one of the ways. Some bacteria can use ammonia or nitrate, and we have tests that will test for that as well. So they can use nitrogen in a slightly different form. And then there are a few bacteria that can actually do what's called nitrogen fixation and use nitrogen gas and actually fix that nitrogen gas into different types of amino acids and proteins. Sulfur we also find in amino acids, and same way we still found nitrogen in amino acids. In amino acids and proteins, you find the disulfide bonds that are made. Thiamine and biotin also contain sulfur. Just like with nitrogen, most bacteria can decompose proteins. We have some bacteria that can use sulfate or hydrogen sulfide, and just like with the SIM test, we looked for the ability of the bacteria to utilize indole. We can also look in that same tube for hydrogen sulfide. We can look to see if it makes this hydrogen sulfide, which is going to be a black precipitate that comes out. It makes the tube look black. Phosphorus we find in DNA, RNA, and ATP. DNA, RNA, and ATP are all considered nucleic acids. We also find phosphorus in the membranes, and we find it in the form of phosphate, or PO4- is phosphate, in these membranes as a major source of phosphorus. So bacteria can use phosphates and break it down and rearrange them put them in various different configurations for the DNA, the RNA, or the phospholipid bilayer. The hydrophilic head of the phospholipid bilayer specifically. Trace elements are inorganic elements that are required in just small amounts. We don't need a whole lot of it, to sustain the life of the bacteria. And remember, as with the physical requirements, not all of these chemical requirements are required in the same amount for all bacteria. They're all different. But for these trace elements, no matter what it is, we need enough of it. We need a little bit of it. And if we don't have that small amount of it, even though we don't need a whole lot, if we don't have that little bit of it, then that's going to be problematic for the bacteria. What we'll find in Chapter 5 is that a lot of these trace elements are usually enzyme cofactors that are responsible for the cell's metabolism. Oxygen is also an important requirement for growth, as we saw. These are what we call thioglycolate tubes, and remember from our lab, with thioglycolate tubes, the concentration of oxygen decreases as we go from the top to the bottom of it. So the first class of microbes that we'll talk about in their requirement for oxygen are obligate aerobes. You'd find them growing at the top of the tube. It means that they have all of the enzymes that are necessary to deal with oxygen in its toxic form, and in fact that they are able to not only neutralize that oxygen, but to use it to make energy. And if they don't have oxygen, they'll die. You, my friend, are an obligate aerobe. Facultative anaerobes are both aerobic and anaerobic growth is exhibited. You'll find most growth at the top of the tube because that's the most effective way to make energy is with the use of oxygen. And when I say energy, I'm referring to ATP. But notice that you'll also find growth kind of growing at the bottom of the tube or throughout it, because these are microbes that can, if there's no oxygen present, they can deal with that as well. So they have some of the enzymes like catalase and superoxide dismutase that will allow oxygen to be neutralized and to use it, but they might not have all of them. At the other end of the spectrum of obligate aerobes, we have obligate anaerobes. So we only see anaerobic growth is going to take place here. So these microbes can only grow where there is no oxygen. they don't have those enzymes of superoxide dismutase, catalase, or peroxidase. They don't have the enzymes that are required to take oxygen from its toxic form to be neutralized as a usable form of oxygen. So they can't tolerate oxygen at all. For aerotolerant, and really in lab, we're just going to be working with these three classes. But as an understanding of the theory behind all of this, we have aerotolerant anaerobes, which means that they can continue to grow in the presence of oxygen. So they will have some, but not all of the enzymes. So they'll have superoxide dismutase, but it can only partially neutralize the oxygen. They don't have catalase and they don't have peroxidase and other enzymes that are required to further neutralize that oxygen. So we do see kind of growth that happens throughout the tube. And then microaerophiles, they have aerobic growth, but they can only stand a little bit of oxygen. So they can't deal with a whole lot of oxygen, just a little bit of it. And so we find those kind of in the middle there. So they can produce lethal amounts of toxic oxygen if they are exposed to normal atmospheric oxygen. So that's why they can only deal with just a little oxygen there. So when we talk about oxygen and its toxic form, a singlet of oxygen can be boosted to its higher energy level state, and it's very reactive. It's not very stable at all. So superoxide free radicals will form when we have this oxygen that's elevated to its higher energy state. What superoxide dismutase does is the enzyme, and I'm going to highlight this enzyme, and I'm going to highlight all these enzymes because they're important. What superoxide dismutase does is it turns this oxygen, this free radical oxygen, into hydrogen peroxide, and then we have just our normal oxygen that's not at this high energy state anymore. It's not so reactive at this point here. The next class of ions that we have, so having superoxide dismutase is a very important enzyme that's required to deal with oxygen if you're going to use oxygen. So if you're going to be a facultative anaerobe, if you're going to be an obligate aerobe, you need to have superoxide dismutase. The next class of enzymes are catalase and peroxidase. What catalase does is catalase will take that hydrogen peroxide, because hydrogen peroxide is still toxic. This guy's still not, you know, something to fool around with. So we take this hydrogen peroxide with catalase and break it down into water and to oxygen, which we can deal with at this point. This is an oxygen that's much more stable. It's not the reactive toxic form of oxygen. And then we have peroxidase that can also take hydrogen peroxide and break it down into water. Not all cells have catalase. Not all cells have peroxidase for bacterial cells, even if it is a facultative anaerobe. If it's an obligate aerobe, chances are that it has both catalase and peroxidase. So there are bacteria that can have all three of these enzymes, or there are bacteria that can only have superoxide dismutase and catalase, or superoxide dismutase and peroxidase, and they can deal with oxygen. Or if you're an obligate anaerobe, they don't have any of these enzymes to deal with oxygen in its toxic form. So I'm not so concerned with you remembering the formulas, if you will, but you should know what enzymes do what. Superoxide dismutase takes that superoxide free radical and turns it into hydrogen peroxide. Catalase breaks down hydrogen peroxide. Peroxidase breaks down hydrogen peroxide. So on to organic growth factors. So organic compounds obtained from the environment, things like vitamins, amino acids, purines, the pyrimidines, the A's and T's, if you will, of or the nucleotides is the appropriate scientific term for this. For the building blocks of DNA and RNA, those are organic growth factors that are required for the bacteria to grow. Biofilms, I think you guys are all very well versed on biofilms, or these bacterial communities. New thing about biofilms that we hadn't quite talked about yet is that they communicate with one another through what we call quorum sensing, so that a bacteria will not make as much of a byproduct as it normally would make if it's going to be toxic to the other bacteria into the community. So that's quorum sensing is really the chemical way that bacteria are able to talk to one another and communicate with one another. And corn sensing also attracts more bacteria to this area and says, please come be a part of our community, come be a part of our little hippie commune here. And biofilms are good for the bacteria. Depending on where the biofilm is for you, not so good, especially if it's on a medical implant like a catheter or something like that, or it's not really good for you. As we said before, they share nutrients and they shelter each other from harmful factors. So now on to our media. These are some definitions here. I'm not going to spend any time talking about them. I'm going to allow you to read them because I feel as though that we have talked about media at great lengths so far in lab, and we'll continue to talk about media and the terms of inoculating and cultures and so forth. So I'm just going to allow you to read that because these definitions are pretty straightforward, and we've discussed this in class previously. So agar is kind of the base for many of our media. It's a complex polysaccharide that most bacteria cannot digest, so it's why it's a great solidifying agent to put in our slants, in our test tubes, and in our petri dishes to allow for colonies to grow on there. It gets liquid at about boiling temperature, and then it solidifies at about 40 degrees Celsius, so that's why it's also great to put in the incubators at 37 degrees Celsius because it's not going to get all gooey and liquidy on us. Cultural media that is chemically defined means that we know exactly what we put into that media. We know the exact chemical composition. We know what kinds of amino acids and how much. We know exactly what kinds of proteins and how much. We're not just saying beef extract, which is just a protein on there. We know exactly what sugars and how much we put on there. So chemically defined media is very, very specific, whereas complex media just means you put a bunch of stuff in there. We didn't really get specific on what we put in there, like the chemical composition of the stuff, but we did talk about we had some yeast extract, some beef extract, maybe some plant extract in there. So we know we've got a bunch of stuff in there, so it's complex. It's got more than one thing in it. However, the chemical composition of each of the entities that we put in, we weren't concerned with that. So when we talk about our nutrient broth, which is the liquid media that we've been using, and the nutrient agar, those are great examples of complex media. So when we look at this table here, notice that we have the exact chemical concentration or the exact chemical consistency of each one of the items that are going into the medium. For glucose, it's kind of standard that we know that C6H12O6, so that's why they didn't put it out in its molecular form. And the same thing with water. We know it's H2O, so why write out in its molecular form? But this is a great example of what a chemically defined media would look like. Whereas for this, this is another chemically defined media because we're saying exactly what salt, exactly what amino acids, exactly what purines, spermidines, and how much of these things we have in each of them. So this is a very chemically defined media. Whereas for this guy here, we got some peptone, partially digested proteins, some beef extract, sodium chloride, agar, and water. So notice that we don't have any of the exact chemical constituents of what makes up the beef extract, so what proteins are there, what amino acids, what sugars are present, if any, what lipids are present. We didn't really talk about all of the things that make up beef extract. We just said beef extract. So same thing with a partially digested protein. We didn't even say what protein it is. So there are several different combinations of amino acids that we could have with that. So with a complex media, there are many different things in there, but we don't know the exact chemical composition of each one of the entities that goes into the media. Anaerobic culture methods are to grow bacteria that don't want to deal with oxygen or that grow best in an area free of oxygen. We can use reducing media like we've seen with thioglycolate tubes, where as you go from the top to the bottom of the tube, the concentration of oxygen decreases. We can even use a process, and to make this reducing media, we kind of drive off the oxygen. You'll not have to do it until you get to grad school if you decide to pursue a degree in microbiology or until you get into kind of your upper level microbiology classes. For us, it's already done. We also can cultivate anaerobic bacteria in a jar with a gas pack here, which is something that we've done very recently in lab. And to get even fancier, we can have anaerobic gas chambers, which you will not find on the campus of St. Louis Community College, but you may see these at some of your four-year schools or at some of the hospitals that you work at, or the CDC, the World Health Organization, places like that that have like higher biosafety labs. They have an actual anaerobic chamber, whereas we just kind of use a jar or we use thioglycolate tubes. Capnophiles are bacteria that like really high carbon dioxide concentrations. We can use a CO2 pack or the old-fashioned way, it's a candle jar that will drive off the oxygen and leave just CO2 present. Biological safety labs, we are obviously a BSL-1. There will be no safety precautions that take place. BL Safety Lab 2, lab coats, gloves, eye protection. We do use the eye protection, but that does not make our lab Biosafety Lab 2. We are still Biosafety Lab 1. Biosafety Lab 2, you'd probably find at more of your four-year universities where they have various different kinds of microbiology classes, not just an introductory class that we have. These are kind of for upper levels that are like virology or medical mycology or things like, different aspects of microbiology. Biosafety Lab 3, they have biosafety cabinets that prevent airborne transmission. They're working with more pathogenic organisms. You would work with organisms under a fume hood. Biosafety Lab 3 are going to be in places like hospitals, so SLU, WashU, Barnes, any of those places. If it has a medical school, it probably has a Biosafety Lab 3, but most hospitals are probably Biosafety Lab 3 for their microbiology labs. And then Biosafety Level 4 is the highest, and they not only have their own circulation in the lab, but the lab is its own self-contained unit. It's completely sealed, negative pressure. The air exhaust is filtered not once but twice, but sometimes even more than that. Center for Disease Control in Atlanta, the World Health Organization, those are Biosafety Lab 4. These are going to be labs that are experimenting to find vaccines for things like the Zika virus or for Ebola or any of these really nasty bugs, these super bugs that we don't want to escape from anywhere at all. So this is the highest biosafety lab level, and these are labs that you don't just foam in and foam out, which means you don't just put the hand sanitizer in your hand and come in and then wash your hands and hand sanitize when you leave out. You have a complete hazmat suit. You're showering to go into the lab. You're showering to come out of the lab. You don't take things home from the lab at all, not even a loop. You don't take anything at all out of that area, and then you have these different chambers that you go through or checkpoints that you go through, and it's kind of the hazmat lab kind of deal that you experience there. And there's a beautiful picture of technicians that are working in a biosafety laboratory number 4. Notice they're working with their organisms under the fume hood, and they even have their own oxygen supply because they are in their hazmat suits here. So different types of media, selective and differential. Once again, I won't spend a whole lot of time on this because we have talked about it in lab. Differential media makes it easy to distinguish colonies of different microbes. Blood agar is an example of differential media. We see that some blood cell or some bacteria can hemolyze or break down the red blood cells, and others cannot break them down. So we have different levels of hemolytic activity, either alpha, beta. Beta is complete hemolysis, alpha incomplete hemolysis, and gamma means no hemolysis at all. Or the differential media that we've worked with are MSA plates, where we saw that either it could break down mannitol and turn the agar yellow, or it could not, but it could still grow there. Selective media will suppress some microbes and encourage others. MSA is also a selective media, where the Staphylococcus epididymis, this is showing a differential aspect of it, that it didn't break down that mannitol, so it didn't turn it yellow, whereas Staph aureus turns it yellow. Selective media, the fact that there are some bacteria that just can't grow on an MSA plate. A lot of gram-negatives can't grow on an MSA plate. It's too salty for them, so it suppresses those and encourages the growth of others. Obtaining a pure culture. using aseptic techniques. A colony is just a population of cells that come from a single cell of attached cells, and we consider that a colony-forming unit. We use the streak price method to isolate cultures and to just get individual colonies to grow, and we've talked about that in previous labs. And this is just one option of streak plate method. What we do for our streak plate method is that we separate into quadrants, just because we're kind of newbies on here, but we streak, flame, streak, flame, streak, flame, streak, flame, so that eventually what we end up with is individual colonies in your third and fourth region, in much the same way that they have individual colonies that are right here that you can pick out. Notice that we had more than one type of bacteria there. We know that because we have these red colonies, and then we have kind of these golden color colonies here. So you could pick up one of these red colonies, put it on its own agar plate, and feel assured that that's all that's going to grow on that plate. That's all that's going to be there. It's that particular species of bacteria. And the same thing for these gold color colonies. You'd probably want to take a gold color colony that is not attached to anything. Like I think maybe right back here, it's kind of hard to see, but it's not directly touching another cell. So you'd probably want to take one of those to put on its own plate to make a pure culture. Ways that we preserve bacteria: we can deep freeze them or we can freeze dry them. We won't be doing any of that in lab, but that's a technique that's used to ship bacteria and how we get them in the mail by freeze drying them or deep freezing them, so that they're not growing and dividing while they're in shipment. The way that prokaryotes are going to reproduce: binary fission, budding, kinetiophores, or fragmentation of filaments, namely for mold and yeast and that sort of thing. Most bacteria that we're going to work with will divide via binary fission, which means that one bacteria is going to become two. And here's just a pretty picture of that. So in theory, these two bacteria should be exactly the same. But what we'll learn in Chapter 8 is that there are various different methods that we can allow for genetic diversity to take place. But in a nutshell, bacterial fission is just one cell becomes two cells. Now, because we understand that, we can kind of extrapolate that data and that idea and that understanding to figure out about how many bacteria are in a generation. So if we have one cell and the numbers are expressed as a power of 2 and that cell hasn't divided, then we just have one cell. So when we look at the visual representation of that, one cell that's not to the power of any divisions gives you one cell. If you have two cells that are to the power of, 2 to the power of 1 gives you 2 cells. 4 cells, the number of cells you have there, 1, 2, 3, 4, because we started off with 2 cells, or one cell that divides and becomes 2, you end up with 4. Another way to think about this is that if you're in the 0 generation, we only have one cell. So one cell hasn't divided, so there are no other cells there. But if we have one cell and it divides and becomes 2, then we have that first generation that's happened. So if we go back to this slide, we can look at that and see that, one cell became 2. Notice how all these cells stayed 2, because one cell becomes 2. Then in that this first one here, that we had this one cell divide in that very first generation and gave us 1, 2 cells. So let me know if you're following here, and we'll do some activities in class with this as well. So if we're in our 5th generation, the 2 stay the same. 2 to the 5th power is 32 cells. So what you're really thinking about is that one cell becomes 2, 2 becomes 4, 4 becomes 16, 16 becomes 32, 32 becomes 64, and so forth and so forth and so forth. So if you have this one cell, after it's gone through 5 generations, so we can use the term generation, doubling, or I'm trying to think of other terms you can use for this. So generation number and cell doubling or divisions, then that's what this superscript number stands for. So 2 to the 5th power is 32. One cell after it's had 10 rounds of division, it gives you 1,024 cells and so forth. So we can just go all the way down the line. Now, we typically work with the log number of cells because it's much easier to graph, and it's a prettier graph, a log number than it is to do 1,048,576 cells. This is going to be a nightmare to graph. Whether graphing this is going to be much cleaner and much more streamlined, so we're just taking the log of this number and turning it into this to make it, just by taking the log of it, the log base 10, just makes it a nicer graphical representation. Now, because we understand that one cell becomes 2, and we understand that we can use the log of those numbers of cells to represent the same data, then we can figure out about how long it takes for a generation to come about, and we can figure out, if we have the right information, how many generations did we start off with. So if we wanted to figure out, if we have 100 cells that are growing for 5 hours and they produce 1.7 million cells, then how many generations did we get in that time, and how long does it take for one generation to take place? How long does it take for one cell to become 2? So what we do here is that we take the number of cells at the end, 1.7 million, and we take the log of that. So as I said before, we don't want to have to actually work with like 1.7 million. That's just like a huge number, and we don't want to have to deal with those kind of calculations. So we can take that number of 1,320,000 cells and take the log of that, which is 6.23. So let me pull my pencil out. And in class, I will show you exactly how to do this on various types of calculators, because you will need to do this for your exam that's coming up here. So the number of cells that we had at the beginning, well, we started off with 100 cells. So the log of 100, I believe, is 2. And it is 2. Now, this number here will always stay the same, and that's because that's just the log of 2. And if you don't believe me, if you know how to do logs, then just do it and type it in your calculator. So the log of 2 is 0.301. And why does it always stay the same? Because one cell in binary fission always becomes 2. So it's going to always stay that exact same way. So now we're just going to do basic arithmetic here. So 6.23 minus 2 divided by 0.301 is going to give us 14. So we have 14 generations that have taken place. Because we understand that we have 14 generations that have taken place, then we can figure out what the generation time is. So we take 60 minutes times the number of hours that we have, which in this problem it says we have 5 hours that are taking place, plus the number of generations that we have, and it turns out to be a little bit over 21. So I think it's like, let's see exactly what it is. Yeah, it's like 21.42, so 21.5 minutes per generation, or 21 minutes to get each generation out there. And as I said before, we will be doing this in lecture class as part of our activities to really get a feel of this, because you will have to do this for your exam. And then this is just a really cool picture that shows you how much prettier and nicer it is to graph the log of something as opposed to graphing the actual cell numbers. So the last thing that we'll talk about are these different phases of bacterial growth. We have the lag phase, and the lag phase is where we're just preparing for population growth. Log phase, where we see exponential increases in the population. And in the stationary phase, this is where the number of bacteria that are entering the population roughly equal the number of bacteria that are leaving it. There are quite a few things that can cause this equilibrium to take place. If you're growing bacteria in an enclosed space on a media that has a finite amount of resources and space, is that you're going to have to, there's going to be this change, there's going to be a stationary point where there just aren't enough nutrients there. And then as the nutrients in the space really start to decrease, then we have that death phase, which is just this logarithmic rate of deaths that takes place there. For serial dilutions, is something that we've talked about as well in class. I'm going to go through it rather quickly because we've done serial dilutions and we've talked about them. But if we want to kind of calculate or approximate the number of bacteria in a sample, then what we look at is that we know that we've taken 1 ml, there's 9 ml of this broth in this tube, and then we took 1 ml and added to it from our original inoculum or our pure culture. And if we just kind of dilute it down, we just kind of add zeros because we're we're diluting it by tenfold, if you will, so that once we get down to this dilution factor here, there is a ratio of 1 out of 100,000 that we see. So to calculate the number of bacteria that are in this inoculum, in this dilution here, we count the number of colonies that we have, and then we multiply it by that dilution factor. So the example that we have at the very bottom, if we have 54 colonies on a plate of a 1 to 1,000 dilution, and then 54 times 1,000 would tell us about the number of bacteria that are in that particular sample. So as I said before, this is something that we've done before in lab and we've talked about, but this is a concept that you need to understand thoroughly, and we'll talk about it again in class because it'll be on your exam. Plate counts, we can count colony forming units. Usually we have a plate counter no more than 250. We can count bacteria by filtrations. These other methods that we can kind of measure the growth of bacteria by counting the number of cells or colonies there. This is direct microscopic count of bacteria cells. So we're directly counting the cells. It's just to give us an idea of how much growth we have in quantitatively speaking using the actual numbers. But we can also use qualitative methods, which means that we don't have actual numbers of bacteria or colonies that we're counting. Instead, we're looking at the amount of growth that we see there. And we can look at turbidity, just kind of eyeballing it and say, it's cloudy. There are bacteria growing in there. It's not cloudy. There are no bacteria growing in there. Or we can use more scientific forms where we can use a spectrophotometer, which will look at how much light is transmitted through a test tube to account for the amount of turbidity. So if we have 100% transmission or 0% absorbance of the bacteria in the test tube because there aren't any there, then that tells us that there's nothing growing. Or we can have something that says we have 20% transmission and 0.70 for absorbance. That means that there are some light rays that have been absorbed from this light by the bacteria, and not all of them are reaching the light-sensitive detector. So this is just a way of measuring turbidity that we just eyeball. We don't have this machine. I don't think we have one, but you probably will get a good chance to work with something like this in an organic chemistry lab and definitely in other microbiology labs. So that is all for Chapter 6. The next time that we have another session, it will cover Chapter 5, and we'll talk about microbial metabolism.